Thank you. When Asim asked me to speak in this conference, I immediately said yes because he told me it was in honor of Golding. And then I looked at the actual title of the conference and it was on synthetic life, and I sort of freaked out. But what I'm going to do is really tell you about some biological systems that we've been using that will help us understand some of this, the processes. And one of the main focuses of my right one? No, that's this one. Okay. One of my main focuses here, as I just said, is to say thank you to Golding. And I wanted to say thank you for a number of reasons. One is that actually Golding was the person who helped me find my first and only job. And the background of this is that I was a postdoc in Cambridge, England, in the lab of Fred Sanger, and Golding came through. This was in the 60s, came through several times, and he and I would talk. And unbeknownst to me, he came back to Madison and talked to Phil Cohen, who was the chairman of physiological chemistry department. And the two of them sort of got together. And this was in the days of the Old Boys Network, I think. And suddenly I got an invitation to come here and apply for a job, which I did, and it was successful. The other reason I wanted to thank Golding is that once I did come, he was a good friend and mentor for the 1 year that we overlapped here in Madison. And afterwards, whenever I would go to Boston, I would stop in and see him as well. And as a consequence of this mentorship, I spent many happy hours in some of the group meetings that Golding had here, and that resulted in a publication. I'm on number 120 of the Studies in Polynucleotides, a famous series. And what I actually did in this paper was to run a pretty messy fingerprint, or a couple of them, and to analyze the RNA transcript of one of the fragments that he was working on. And when I was looking at this publication this last week, I recognized that this is a 29 nucleotide unit, and it's just too bad that Golding didn't know about microRNAs at the time. It could have saved him a lot of work in synthesizing the first gene. So, since that time, I've continued to collaborate, to work on RNAs in a number of capacities. And during Masiasu's introduction this morning, it occurred to me when he said that Golding always liked to change geography once every 10 years or so for stimulation. It occurred to me other people change spouses once every 10 years for stimulation. And I often change collaborators. And I've been able to, privileged to collaborate with Václav Cybulski, Fred Latner, Dick Burgess, and of course Masiasu. It's been wonderful. That's one of the great advantages of being here in Madison. What I'm going to talk about today is collaboration with Mike Sheets and Elspeth Lund here. And this is sort of a follow-up on what Phil was just talking about, although I'm going to be talking about a different function of microRNAs and a different system. The system we're working on is the early embryos of Xenopus laevis. So this is a little bit of developmental biology, but not a lot of development. But it is a biological system that we've been using. And as you already heard from Phil, microRNAs will form imperfect hairpin, imperfect duplexes with a targeted sequence in a messenger RNA in the presence of an agoprotein. There are several different forms of agoproteins in cells, and some of these oncoproteins will recruit other proteins. In the case I'm talking about here is a protein called GW182, this in turn will recruit other proteins that can lead to deadenylation of targeted messenger RNA and subsequent degradation. And in contrast, small interfering RNAs come from perfect duplexes generally, but they form perfect duplexes in messenger RNAs. And one of the oncoproteins, OGO2, that Phil also mentioned, has this activity. It's an endonuclease activity known colloquially as slicer, which will form a break in the RNA and degradation. And here's the beauty that we work on here. And I'm only going to be talking about events in this very early cleavage embryo stage of the frog development. In cleavage embryos, gene expression depends on maternal RNAs and not on zygotic messenger RNA synthesis. And at this stage of life, these little embryos are doing everything they can to make more DNA, to make more cells, but they don't make any messenger RNA. They depend on messengers that were stockpiled in oogenesis. At the mid-glacial transition, after about 8 hours of cleavage, these cleavages, cell divisions are about every 30 minutes. At about 8 hours, the zygotic messenger RNA synthesis starts. The cell cycle is remodeled, slows down, and a lot of the maternally generated messenger RNAs turn over. And that's what I'll be talking about in the next couple of minutes. You already heard this about this pathway. In the case of early embryos, one particular microRNA, 427, accumulates very high amounts. By about 8 hours, we detect about 10 to the 9th molecules per embryo distributed throughout the embryo. And the reason that the embryos are able to make so much of this is that the genes encoding miR427 are highly reiterated in the genome, and so there are lots of copies of a primary transcript that are processed. In the embryo, the oncoproteins associate with these RNAs, and we have what is called a RISC, RNA-induced silencing complex. We also have short-circuited this whole process. One of the advantages of using the Xenopus embryos is that you can generate these premiers in vitro by just in vitro transcription and inject them into fertilized eggs. And you can see now the accumulation of miR427 much earlier than it would occur normally. And in fact, it also allows us to put into this system exogenous microRNAs that would not otherwise be present in the system. Now, the other half of the players here, the targeted messenger RNAs, and in this case we have cyclin B2, just as an example. This is the northern block. This is the endogenous RNA. You can see as a function of time after fertilization, this RNA becomes destabilized, and at about mid-glacial transition is when it really starts to disappear. Rebecca Hartley, a collaborator, showed that there is a region in the 3' UTR that is essential for this degradation or this destabilization, and she showed also that the destabilization requires zygotic transcription, RNA polymerase II. And this was before the days of microRNAs, and it's just sort of stood as an unknown. But what we think is happening here is that it's miR427 is involved, just to jump ahead. So the degradation of this particular messenger RNA occurs through deadenylation of the messenger. The analysis that we did is just shown here schematically. this molecule is too big to see a significant difference by degradation of, or removal of the poly A, a difference in its mobility. But if we just cut a little bit off that would contain the poly A and run that on an acrylamide gel, you can see that here the RNA gets much shorter, just at mid-last of the transition. If we block miR-427 with a complementary oligonucleotide, the messenger RNA is stabilized. And conversely, if we introduce into these embryos miR-427, as I just described earlier, we can accelerate the onset of the deadenylation. So we conclude that miR-427, in fact, does promote this deadenylation. Looking at the sequence in the cyclin B2 message, we can see a region that is complementary to 427, and Phil emphasized the importance of the seed sequence, and there's a perfect match for the seed sequence here. And this is just showing that in this case we have a reporter RNA where the beta globin coding region is attached to the 3' UTR of cyclin B2, just make it easier to assay. And we can see if this is injected into the embryo, the RNA becomes polyadenylated and then decays upon deadenylation. Now, what we've just to test this interaction specifically, we've mutated part of the seed sequence, and when we do that by changing CAC to a GUG, it no longer is perfectly complementary to the microRNA, and the RNA is stable. And in the experiments that I don't have time to show, we've been able to make a compensatory miR-427 that would have the complementary sequences in the microRNA, and now we get restored the deadenylation. So we conclude that the 427 really is important here. Other experiments that I don't have time to talk about are just listed here. We can put in an entirely different system here. Instead of using 427, if we put in say a LET7 target into the microRNA, into the messenger RNA, and then express LET7 microRNA in these embryos, we again get deadenylation. So it's not a sequence specific, but it's a match specific interaction. We also have shown that translation is not required, and if we move the target into the coding region, it works, but it's very inefficient, probably because ribosomes come along and knock the complex apart. Finally, I want to emphasize that the target, we've been able to show that the target is really quite small. It includes the seed sequence and a few adjacent sequences as well. What's interesting in the two cases that we studied in detail, the cyclin B2 and A1, there's only a single target in each of these messenger RNAs, as opposed to the multiple targets that one often sees in 3' UTRs for microRNAs. A single target, and that one target is both necessary and sufficient for deadenylation. So the ability to get along with a single target might reflect the high concentration of the microRNA. So where are we here? I've shown that the embryos are an excellent system for studying microRNA function, at least for this particular function. It's an in vivo system. The components can be introduced in a synchronous manner, and that allows us to make deductions about precursor product relationships that might otherwise be very difficult to establish. We can generate targeted substrates and test them very easily, just by injection. The products are easily isolated and analyzed, and finally, we are studying something, a real organism that is really developing and is really necessary for the frogs, if you want to make more frogs. Now I want to change over to talk about siRNAs. These are the perfect matches to the messenger RNA. And I'm going to tell you something quite surprising to us, that the Xenopus embryos are unable to carry out RNAi, the destruction of messenger RNAs through the mediated by the siRNA perfect complementarity. And also I'm going to say something about unexpected activities of siRNAs in these cells. Now, the experiment that we did here was to move into the coding region of this reporter RNA a sequence that is a perfect match for miR427. So we're just going to use the microRNA that's endogenous in these embryos and have it direct AGO2 to the targeted sequence in the messenger RNA and see whether that will degrade the RNA. We can either use it in endogenous or make an siRNA of the sort that Phil mentioned also. And strikingly, you can see that this RNA, over a very long period of time, is stable. This was very unexpected. It means that these embryos are missing something that is necessary to carry out RNAi for the cleavage. And the obvious candidate for what might be missing would be the AGO2. And so when we put AGO2 into these embryos, we can restore the cleavage. We also see that the slicer activity of this AGO2 is not necessary or is necessary because it's the endonuclease activity that's working here. In this system, we can also isolate and analyze the two fragments that from the cleavage. And so this shows that the early embryos are deficient in AGO2 activity. Endonuclease. How much time do I have? What? I'm done. Well, basically, what we have now shown, and I will not show you the data, is that the importance of having AGO2 in the embryo is that it can somehow counteract the inhibition of the siRNA that's been put in there and allow the embryos to carry out microRNA generation. And let me just pull down to the end here just to say that we can see the generation of 427 only in the presence of added AGO2, and so we know that the microRNA processing is defective when you put in a great amount of the siRNA into these embryos. And with that, I think I'll stop. Thank you.